All right, today we're diving into something pretty wild, a hidden talent of one of the most stubborn cancers out there. Turns out it has a sense of touch that it uses to stay alive. So, yeah, let's get right into it. It really all boils down to this one question, right? A question that's been bugging scientists and doctors for decades. Why is pancreatic cancer so incredibly tough? Why is it so good at shrugging off our best treatments? And the answer? Wow. It's way more complicated than anyone really thought. Okay, so section 1, a stubborn cancer. And look, to really get this breakthrough, you first got to respect the opponent. Pancreatic cancer isn't just aggressive, it's smart. It's sneaky. The real heart of the issue here is just how adaptable these cells are. I mean, think about it. You hit them with chemotherapy, and instead of just rolling over, they change their entire game plan. They don't just die, they literally shift their whole biological state just to survive the attack.
So, how in the world do they pull that off? Well, it's like they have this built-in biological survival switch, and they can just flip it anytime they feel threatened.
And that switch, it's a process called autophagy. The best way to picture it is like a cellular panic room. When things get really bad, like chemotherapy bad, the cell literally starts eating parts of itself. It recycles its own components just to get enough energy to stay alive. It's a totally desperate move for sure, but man, is it effective. So let's just walk through that real quick. First, the cell senses a threat, right? It immediately kicks on this recycling program. And what that means is it completely stops caring about growing and dividing. All of its energy gets poured into one single goal: just survive. And this is why it's such a huge problem for us. See, most of our chemotherapy drugs, they're designed to hunt down and kill cells that are dividing really, really fast. But the second a cancer cell flips that autophagy switch, it stops dividing. It just goes dormant. So it basically puts on an invisibility cloak, hiding from the very drugs we're using to try and kill it.
Okay, so for the longest time, the thinking was that this switch got flipped mainly because of a lack of food, nutrients. But this brand new research, it's pointing to something totally different. A new trigger that has everything to do with the cancer cell's sense of its own physical surroundings.
And I mean, this is a huge shift in thinking. We've gone from, oh, it's about food and energy, to, wait a second, it's about what the cell is physically touching or not touching. That physical context is actually a massive trigger. This environment has a name. It's called the extracellular matrix, or ECM for short. The easiest way to think of it is like the scaffolding of a building, or maybe even the soil that the cells are growing in. It gives them structure, it sends them signals, and it turns out these pancreatic cancer cells are incredibly sensitive to it. So how does this sensing actually work? Well, for a normal, healthy cell to grow, it needs to be anchored to this ECM. So if a cancer cell finds itself just floating around, unanchored, it completely freaks out. It uses this special protein on its surface called integrin alpha-3. Think of it like a tiny fingertip to constantly feel for that ECM anchor. And if that fingertip can't feel anything, bam. That's the signal. Time to flip on the survival switch.
And listen to this. The lead author of the study, Dr. Mohammed Assi, he puts it perfectly. He says, the cells are basically making a strategic choice based on where they are. They're asking themselves, am I anchored and safe enough to grow? Or am I floating free and I need to hunker down and just survive?
This whole discovery leads to something, honestly, kind of mind-blowing. A single tumor isn't just one big blob of the same thing. It's actually more like a city, with two totally different populations living right next to each other. Yeah, inside the very same tumor, you've got two groups of cells doing completely opposite things, and the only thing that determines their behavior is how close they are to that ECM scaffolding. Okay, so this table really breaks it down for us. Look at the cells that are right up against the ECM. Their autophagy level is low. They're in growth mode, dividing fast, proliferating, and because of that, they're vulnerable to chemo. But now look at the cells that are far away from the ECM, the ones that are floating. Their autophagy is cranked way up. They're just surviving, not growing, and guess what? They're totally resistant to the chemo. And this right here, this explains so much. It's why drugs like hydroxychloroquine, which are designed to block autophagy, often don't work that well on their own. Sure, they might kill off those resistant surviving cells, but they do absolutely nothing to the fast-growing ones. You're basically fighting with one hand tied behind your back, only going after half the tumor at any given time. But this is the beautiful thing about science, right? Once you truly understand a problem, you're halfway to solving it. And this new insight, it doesn't just lay out the challenge, it shines a spotlight on a brand new weakness we might be able to exploit.
So, what's the new strategy? It's actually pretty brilliant in its simplicity. Instead of just another brute force attack on the cancer, the idea is, what if we could trick it? What if we could jam its sensors, essentially blindfold those little cellular fingertips so they can't feel anything?
And that is exactly what the researchers did in their experiment. They went in and genetically shut down that integrin alpha-3 sensor. And the result was incredible. All of a sudden, every single cell, even the ones touching the ECM, thought it was floating free. So they all panicked. They all flipped on that autophagy survival switch. And just like that, the tumor wasn't divided anymore. It became one uniform target, with every last cell in the exact same vulnerable state. And the result of making that tumor uniform before attacking it? Get this. A 50% reduction in cancer cell survival. That's huge. Just by forcing all the cells to rely on the same survival trick, the drug designed to block that trick became way, way more powerful. And they're not stopping there. The research is already moving on to the next step. Now they're looking at a protein called NFR2. Think of it as the internal messenger that relays that, uh-oh, we're floating, signal from the sensor to the rest of the cell. The idea is, if you can disrupt that messenger, you might have another, maybe even better way to shut down this whole survival system. So the key takeaway here is pretty profound. This is way more than just discovering some neat biological quirk. It's a completely new way to think about treatment. By messing with how a cancer cell literally perceives its own world, we can basically herd all the cells into the same trap. And that makes the drugs we already have so much more effective. And that leaves us with a really fascinating thought to end on. We've just seen how powerful it can be to target a cancer's sense of touch. So what's next? I mean, could we start tricking how they sense nutrients or oxygen or other signals in their environment? It really feels like the whole fight against cancer might be shifting into a battle over perception itself.